Welcome to the Wizards of Biobanking podcast. I'm Shrikant Adiga, CEO of OpenSpecimen, and I will be interviewing biobankers from different backgrounds and countries to learn and explore the world of biobanking. Hope you will enjoy this interview series as much as we enjoyed producing it. Thank you. Hi, Louise and Ashlin. Welcome to the Wizards of Biobanking podcast. This is the fourth session we are doing with different biobankers across the globe. So the intention here is that we talk to you guys to just understand the industry, understand how you work and what you do and how it impacts research, and also make it a repository of collection, because every biobank is different, every collections is different, people work differently, operate differently. So it's a good repository of someone starting a new biobank, or want to see something, learn something. Then it's a good repository for people. So that's the intention. Thank you very much and welcome.
Thanks, Ray.
So with that, so I'll let us first start with the brief introduction of both of you, what you do, what is your background, and how did you end up in biobanking?
Sure. So my background is as a biochemist and a cancer biologist, probably for more than 25 years. My research has really been around looking at transcriptional networks to do with cellular differentiation and commitment. And probably about 5 or 6 years ago, there was an opportunity within the cancer division at the Harry Perkins to build a translational pipeline. And this was something that I was really passionate about. And as a researcher, I really appreciate the need for it and really understand that it is a really important part of our research. So the biobank was part of that pipeline. So I said I agreed to be part of it and to start it off, and I've ended up actually seeing it all the way through to its initiation and stayed with it, which has been really quite amazing. And I think, it's grown incredibly quickly. We've probably got more than 5,000 samples in there currently. And a lot of that has been driven by a partnership that the Biobank's now got with the Liver Cancer Collaborative to collect biosamples for them. And I guess this is really, having such a lot of samples come in so quickly, one of the things we've been really mindful of is that we have the appropriate digital environment to monitor and to manage all of these samples. And this is where Ashlin comes in. So we engaged Ashlin probably a year ago now, 18 months to take on all of that role. So I'll pass over to Ashlin.
So I have a background in computer science and IT. So before joining Perkins, I used to work for some data scientists, and I basically looked after their IT infrastructure and the data, helped them collect the data, store it, build tools to analyze it. And since joining Perkins, I've been basically doing the same thing here at Perkins. And so looking after OpenSpecimen and the DRE, so messaging all the data to get it in and move from one to the other. And
yeah, that's great. Having someone like Ashlin up front and available also meant that before you guys even talk to us about OpenSpecimen, Ashlin was already an expert in OpenSpecimen. He went into the demo site, figured out most of the things. So that kind of helps a lot in such setup, especially when you're just starting out. There are a lot of things to explore, and someone there to at least take care of the IT problem is good. Okay, so moving on. So let's talk about your biobank. The Harry Perkins Center is part of University of Western Australia. How does the whole setup work?
Yeah, so the Harry Perkins Institute of Medical Research is affiliated with the University of Western Australia. So we're a medical research institute, but with that affiliation. We are based on one of the tertiary hospital campuses in Netherlands, close to the university, but embedded within a hospital campus, which has been really instrumental, I guess, in the way that we've gone about building our biobank. And I think we were lucky because we were building it from scratch, I think, and that really allowed us to set it up the way that we wanted. And coming from a research background, I really had some very clear ideas as to where I wanted it to go and how I wanted it to be established so that it would really provide the biggest benefit to the researchers. And so we spent a lot of time building the governance structure around it. I spoke to the Human Research Ethics Committee for the hospital numerous times, backwards and forwards, with various iterations of our governance document before we submitted it. And what we've created really, I guess, is a governance structure that allows us to collect samples, so we can consent the samples, consent the patients, collect the samples, process them, and then distribute them back to various technology platforms or researchers to generate the data. But then we can also collect back into the biobank any secondary samples that are generated. So, for example, cell lines or organoids or PDXs. And also capture all the data that's generated from those research outputs. And that was really the key, was that we could, rather than just having a biobank of biospecimens that you give out to people and then they disappear, we're trying to build a database with it. So all of the data that comes from that research, we try and capture, and then it becomes available to the entire global research community. So that was our goal from the outset. And the other really key part about that was that there wasn't a lot of having to reapply for ethics every time there was a different project that we wanted to do. So we made it very broad across all of the research activities that we were doing. And we also, and because it was so broad, we were very careful about speaking at great length to our consumer engagement and community engagement panel, just to make sure that it was not something that they would think would be an issue. And they were all very supportive of it. So we went ahead with it. And it did take a while to get that type of governance and ethics through, but eventually, I think probably about 3 years backwards and forwards to get the ethics and then the governance at each of the hospitals. So it was a pretty long process, but we've ended up with a really, I think, quite unique situation, and it's really facilitated some amazing research projects. The other thing that was really important with the biobank was that we, because we needed to manage the samples so carefully and across so many in and out of the biobank, we needed a really good laboratory information management system. So, which is why we chose OpenSpecimen. We spoke to a lot of people and we looked at a number of different products, and that seemed to be the one that would be able to really manage what we wanted to do the best. And we were lucky to get a grant from the Western Australian Department of Health to build that OpenSpecimen into our biobank. And we're also building a separate platform at the other end of the biobank to capture all the data. And this is a really amazing product that we're working in partnership with a company called Iridia to build this digital environment.
And you mentioned about the liver cancer collaborative. Are all your specimens around liver cancer, or do you collect for multiple diseases?
Yeah, look, we do collect from a number of cancers, but and it's really based on the funding that we get. So it's a lot of prospective collection at the moment. And so if researchers get funding through our National Health and Medical Research Program or through the Medical Research Future Fund, that those projects then can leverage the biobank to collect the samples and provide them with the samples to do the translational work that they need to. But we were very lucky almost coincident with the gaining of our ethics and governance, the Liver Cancer Collaborative in Perth was awarded a Cancer Research Trust grant of about nearly $11 million. And a really big part of that grant was to set up a biobank of liver cancer samples. And the really key to this whole process was that we were working very closely with the clinicians. So as well as getting really longitudinal cancer samples, we could also integrate that with linked clinical data. And that was really important to this whole program. And I think in terms of what we're doing with our biobank, that really is something that we want to be able to do.
Yeah, that's interesting. So let's dig a bit deeper into your specimen collection process, for example. And every biobank is different. So I that is why I want to understand a bit deeper. Where let's start with, say, the specimen collection, and before the specimen collection, enrollment of patients. So are patients enrolled by the biobank, by the researchers? Are you involved in that process?
So we work very closely with the clinicians. We've come to the point where we know and we really understand that unless we work very closely with them and they really direct patient consent, we just don't get get access to the patients. So whenever we have a new cancer, for example, that we want to start collecting, we set up a group that involves researchers and clinicians. With the liver cancer collaboration, we've been really lucky in that because it's so well funded, we've been able to have research nurses at every hospital as well. So it's a mammoth effort to coordinate all of that collection of understanding when the patients are coming in, which ones we want to collect, consenting them, and then collecting the actual samples. So it's really coordinated through each of the research nurses, and they're invaluable really to us being able to collect these samples. One of the real issues in Perth, I think, is that the hospitals actually have quite separate IT systems, so it's very hard to monitor across all of the hospitals. So we found that it's best to have, for us, it works better with a research nurse embedded in each hospital, and that's worked incredibly well. And I think it's something that we really has benefited or really enhanced our ability to collect so many samples so quickly.
So you mentioned each hospital. So how many hospitals do you work with?
So we work across the 3 main tertiary hospitals in Perth. So we have 3 big hospitals, one in the south, one in the north, and one east in the centre of Perth. We also do have ethics across some of the private hospitals as well. At the moment, we collect some samples from those hospitals, but not as much as from the teaching hospitals.
And you talked about the nurses in this hospital. Do they directly register the participant in OpenSpecimen, or you guys do that? Do they have access to the system?
They do. So some of them do. We sort of try and do it here where we are, where Ashlin is really, because this is something that he directs. But we provide a de-identified biobank number to the nurse prior to collection, and then they will then send the sample back to us here, which is where we physically store it, and it gets entered into OpenSpecimen. And I'll let Ashlin talk a bit about how we do that, because that's his expertise.
So we've actually got it's quite handy to have like a proper collection form, which we've actually worked with the nurses to try and do, because looked into try and use OpenSpecimen like then and there, put straight in, but it just wasn't working between different hospitals and trying to work for like the speed of the collection as well. So they've got a form we've worked quite closely with to try and collect everything we need and trying to reduce the options for errors. And at a later point that goes into OpenSpecimen. We're trying to line up the fields up as well.
And that's more of a paper form.
So it's an Excel spreadsheet. So when they're collecting, they have all of our tubes, we collect into barcoded tubes, and they have a scanner and they scan. So mostly we're either collecting into liquid nitrogen, so we snap freeze in theatre. And as they're doing that, they will scan the tubes and then into an Excel spreadsheet, which is our collection sheet. And then they'll all be transported down here, and we'll just pull out the collection sheet and then transfer the data onto OpenSpecimen from that. I think that's probably because we only really physically store the samples at Perkins.
So these tubes do you provide those tubes? Do you pre-barcode and give them the tubes, or how does that work?
The tubes are already barcoded, so they just get entered straight in as they are.
So you don't prepare kits?
Sorry, we don't need to barcode them. They come pre-barcoded.
Right.
Okay.
So regarding patient enrollment as a biobank, do you put any efforts in publicizing, helping the researchers in enrolling patients or identifying the patients for which study they can fit in and things like that, like surveys or things around that?
We don't really do surveys and things, but we do that. We are very much involved with setting up a collaborative group with the clinician and the researchers, and then following up with the clinicians to identify patients that they think will be appropriate. They have all the forms, so we provide them with all the consent forms after we've sort of met with them and discussed with them and found if they're interested in being part of a collaborative study. And then they will just let us know when there's one coming, or the nurses, and we'll look through the various databases that they have in terms of for the surgery at the hospitals, and they might go to the surgeon and or the clinician that's in charge and just say, do you think these people will be appropriate? And then the surgeon will of course always make the decision, or the clinician, and then they will collect the actual physical sample after they've been consented, and coordinate transport back to us here. So it's really driven by the clinicians, the actual collection of samples and who's appropriate. So they see all the samples in their all the patients in their clinics and or at an MDT meeting for the liver cancer. I know they have they have MDTs every week that have it, and they discuss all the patients and decide whether or not they will be appropriate for the biobank. And then the patients that will be indicated to the nurses, and then the nurses will coordinate the collection.
So you talked about consenting. So do you do a broad consent, or is this very specific consents per study?
No, the consent that we have is we use a very broad consent. Occasionally, for some of the cancers, we have changed it a little bit and just added a few things in it. And generally, that's around the actual physical collection process, because sometimes it differs for different cancers. So some are done through biopsy and some are done through surgery. And so that may be changed and be a little bit be cancer specific, but we have a very broad consent form that where we collect samples that are access to diagnostics. So we don't. So there's always a sample that will go for diagnosis first, and then we can then get what's left after that, and we will we'll collect those samples. So yes, the consent is very broad in terms of what might happen to the sample after it's been collected. Which is fantastic for all of the researchers. They're very, they're really pleased that it's been set up like that.
So let's talk a bit about what happens to the specimens after they are collected, and how do you maintain the physical quality and safety of the specimens in terms of their storage? What kind of are there any certifications that you are already done or aiming for? General biospecimen quality is of big interest for everyone, right? So how do you handle that?
So we collect two types of samples, really, I guess. We either snap freeze them immediately in theatre, so that really allows us to maintain the integrity of the sample. And then it goes into a -80 freezer at the site it's collected until it can be transported on dry ice to us here. And there are couriers that are going between all of the hospitals very regularly. So we can usually, unless it's a very late collection, we'll transport it, within an hour or two. And then it goes in and it gets entered into OpenSpecimen and our freezers here at Perkins. The other type of samples we collect are fresh samples, and this is when we might want to do something like grow organoids or do single cell sequencing. And these samples are collected on ice into media that's provided by the researcher that's going to do the actual assay. And again, they're collected on ice and transported immediately via courier to wherever those researchers are. So they may be here at Perkins or down at one of the other universities that are part of the collaboration.
And in terms of storage, is it stored? Do you have an on-site storage location, or is it off-site? And how do you manage the inventory?
Yeah, so it's on. We have on-site storage, which is and it's -80s and liquid nitrogen, and it's all monitored and alarmed. So we know at the minute anything happens, if the temperature goes down above what it should be, we get notified immediately, and someone will come in and sort it and see what's going on.
You talked about patient enrollment, collection, how do you process it, etc. Now, the last part is researchers, right? So giving the specimens back to research, how does that process work? So are these specimens meant for specific researchers who get it, or do you have a collection where people can request for specimens later, and then you will review that request and distribute the specimens?
Yeah, I might let Ashlin talk about that, because that happens in the IT at the other end of the process in the DRE. So Ashlin, I'll pass it over to you.
Yeah, so I think we kind of cater for both processes. So we've got the fresh tissue that's kind of requested beforehand that can go straight to the researchers, but then all this other all these frozen samples and blood samples and everything, they're made available. So researchers can come and look through inventory and select what they need and apply for it. So we've one of the key features of OpenSpecimen I'm interested in is actually the dashboard, because it's important for us as part of this project to actually make our inventory available to the public. So we're in the process of setting up a dashboard and putting it on our website so people can actually come to our website and see what we've got. The other side of that is in our DRE as well. We got all the information about our samples, and as well as some additional clinical information, so people can actually look for samples based on either etiology or what data is available, and then enter their requests for the research.
I was just saying we're working on setting up some workflows from the DRE. We haven't quite completed those yet, so we're still sort of, it's a bit of a hybrid model at the moment, where we can work within the digital research environment to find and request the samples, but then it sort of becomes an email after that. Yeah, and because the other thing is because the governance of the biobank means that people have to sign agreements to provide the data back into the DRE at the end. So there's always some involvement with one of us to make sure that that happens.
So, you talked about the dashboards that you want to build in OpenSpecimen and publish it on your website so that people can see what kind of data you have. The other aspect of dashboard is that you can also build an online catalog where people can actually see what kind of specimens you have in the repository. Are you exploring that also?
Yeah. So we're part of it is the dashboard, and yes, setting up the catalog as well, so they can see aggregations of what we've got.
Right. So with that process, they can actually add specimens to the cart, submit a request, fill in a request form, and if they need to upload an agreement, they can also do that. So that whole process can be taken care of there itself.
Yeah.
Okay, so we talked about the specimens and things like that. So let's move on a bit about a few other things. So the one of the things that is of prime interest to biobanks is sustenance. Either it's centrally funded or you have to charge your researchers for use of specimens or storage of specimens and things like that. So how does that work for you? And what are the things that you thought works well in this kind of environment?
Yeah, it's a very difficult thing for biobanks globally, I think, and it's a real problem. And I think when we were considering this, there wasn't an opportunity to get a big grant. It was a small biobank. So we sort of decided that when we were going to set it up, it was going to be prospective collection. So it's pretty much based on who has funds to provide it. So Perkins provides some funds to help run, and that they house it all. So they're paying all the infrastructure costs around housing the biobank. They also provide some salaries as well. So they've been very generous in setting this up. But generally, in terms of what we collect and how we fund that, it's really about who has been successful with grants and things. And then we'll focus on that particular type collection, those particular samples. It's very much researcher driven and always has to be in collaboration with a clinical team as well. So, our funding comes from a lot of different places, but still the samples that are collected will be stored in the biobank and are available to either be used collaboratively with the group that is using them, or, if there are other samples available, then, they can be requested by the rest of the research community. We also do try and apply for any grant funding that becomes available. So there are some infrastructure grants at the moment in Australia that we've been applying for. And that will just help provide, the infrastructure that we need to really make this available to everybody and cover off on all of the data collection and capture.
Yeah, so one of the senior researchers in US told me that the golden rule of research is that the one who has gold rules. So, so that's the golden rule.
Unfortunately, it is a little bit like that. But having said that, one of the really important things I set out to not do with this biobank was end up with a biobank full of samples that sat there for the next 30 years, because we have so many of those around the world. So I was very keen to make sure that the biobank gets used. And one of our researchers said to me the other day, she said that, you know, the best biobank is an empty biobank, and she's absolutely correct, because that's what we do it for. And so being able to then capture the data is such an important part of this biobank. And really, you know, all biobanks should be doing something like this, because that's how we share this biospecimens, really, isn't it? Through sharing the data.
Right. So, you know, while this, you know, how the quality of the specimens and what kind of data is associated with the specimen becomes important.
Absolutely. So there's all sorts of research data. So at the moment we're doing a lot of omics. So we're doing for the liver cancer, which is probably the project that we have the best funding for. We're doing whole exome sequencing and RNA sequencing, some single cell sequencing. We're also generating organoids from as many as we can to do some high throughput drug screening. And we're also collecting PDX, making PDX models in mice. So there is a huge variation of data types that are going to have to come back in. And I might let Ashlin talk about this, because he's probably in a better place to talk about it than I am.
Yeah, so collecting the clinical data, it's been a huge challenge for us. Actually, there's just so much we're trying to do. So as well as, you know, collecting all the data, we're also trying to build a model of, you know, not just like the patient's journey, but for the liver cancer project, because liver cancer is so heterogeneous, we're kind of doing the study at a tumor level, as it were. So we're trying to tie back all the, you know, all the specimens, the treatments, the blood tests, and tie them all back to the individual tumors, because quite often that a patient will have more than one of them. Yeah, so trying to build a model of that in OpenSpecimen, and then transform that into our DRE, so that all our researchers can actually, you know, access and use it, has been one of our current challenges. We also want to you know, make sure that, you know, all this data is accessible. So in our DRE, the data, we try to achieve the FAIR principles of making sure that, you know, everything is findable, accessible, interoperable, and reusable, because it's like, you know, it's there to be used, so we need to make sure that it really can be.
Yeah, that's amazing. So now, you know, let's before we move on to the last few questions, let me ask you a few questions about Perth. So have you been in Perth all the time until now, or are you a Perth?
I was born in New Zealand, but I have been in Perth probably, well, I don't know how, a very long time, probably maybe nearly 40 years. So yeah, so it's been my home for a long time. And I absolutely love it. I mean, if I could, we thought we might try and have the sky in the background, but it's so sunny and bright, it just totally washes out. So we're staring at a bank of windows, looking out at beautiful blue sky, down to the river with boats sailing on it. It's absolutely magnificent. You should come and visit. Oh, you have?
Yeah.
Yeah. But you know how lovely it is. Yeah.
I'll come when there is a cricket match between India and Australia in Perth.
Yeah.
at the Wacker, right?
No, so, it's no longer. We have a new oval, the Optus Stadium.
Yeah, Optus. Yeah.
It was the Wacker for many years. But yeah, no, that would be fun.
So what about Ashlin? What's your favorite thing about Perth?
I've been in Perth my whole life and grew up in the hills. So I actually really enjoy just how close it is to you know, to the forest. It was, you know, 20 minutes to the city and 20 minutes to bush track. So it was it's really nice. I think it's the best of both worlds.
And do you follow cricket?
No.
So coming back to biobanking, we covered a lot of things. Let's move on to what are the bigger challenges in terms of both biospecimen science or in general biomedical research that you face in the realm of biobanking?
Yeah, look, I think, there are some very practical challenges around the physical biobanking, particularly with the way we've set it up in terms of making sure that we get back into the biobank all of the secondary samples that belong in there. So those are things like cell lines and organoids, and as soon as you pass a sample over to a researcher, they get so excited about having a patient sample in their hands, and they want to do so many different things with it. So we've got to be careful about managing that side of things, and OpenSpecimen is fantastic for that, because it gives us all of these audits, and we can put all these queries in, so we just get lists of things that tells us exactly where every sample is, so we can follow that up really easily, and that's amazing. In terms of I think that's probably the main challenge for us. I mean, one of the things we really want to do with this biobank and our digital research environment is generate precision medicine databases and really provide something into the clinic that's going to be useful for the clinician. So that's really the end goal of a lot of the research that we're doing, and really is driving, the real efforts that we're putting into capturing all of this data. So yeah, it is a challenge, but it's definitely worth it. And having a really committed team of researchers and clinicians really makes it a lot easier, and really will really enable us to get to where we want to be with this, and what we think is, what we're trying to achieve.
Right. So the next question is about OpenSpecimen. But before we get to OpenSpecimen broadly, I think it's been a year or so since you guys got onboarded on OpenSpecimen. But before that, if you're a new biobank and you are trying to finalize your IT platform, what are the things that one should look for before they decide on one or the other? So what is the process you followed, or what did you learn from that process? As this is what.
Yeah, it was pretty tricky actually. Initially, we were looking. Because we wanted to collect so much clinical data as well, because that's really critical to developing any sort of precision medicine tool. We needed something that would be able to collect that, but we also needed some sort of IT that would be able to follow the sample through all of its generations and all of its transformations, so that we knew exactly, we could have a really good audit trail of where the sample started and what it ended up as, and where it went, and what research data was generated at what point. So that was because that was our goal. We tried to find something that would cater to all of those needs, and really OpenSpecimen was really the only product on the market that could do that. I might let Ashlin talk about how we're using it with the clinical data and how he's setting that up, because that's a really important part of it. And it does have some challenges. So I'm going to let you talk about that.
Yeah, so I think we're making a good use of the forms, and I think that's quite important, because I know you can have programs like REDCap to store everything done separately. But because we were kind of building it from scratch, we didn't have an existing database. So I think the ability of just putting everything into OpenSpecimen, it's all in one place, and you can tie everything, directly back to the participant has actually been, it's made everything much easier. And it's really simplified the process of also, it's one place to put it in, one place to put it out. It's yeah.
Can you name a couple of forms that you created? Like what kind of data do they have?
So we've got dedicated forms for the like the primary cancer condition, as well as the individual tumours. We've got treatments, whether it's surgical treatments, radiotherapy treatments, local regional treatments, systemic treatments, comorbidities and etiology, and all the related diseases people have that can lead to liver cancer. And as well as questionnaires as well, just on lifestyle and all that.
Wow, great! That's quite extensive.
Yeah.
And that's the intention of the forms module, to enable each project to decide what they want to collect and be able to have everything in one place. Because finally, when you're looking for specimens, you want to be able to say, okay, these are the clinical parameters, these are this one, and then do I have specimens for that? If you have data spread across different databases, Excel sheets and things like that, the whole process becomes very time consuming.
Yeah, and it's been tricky to try and get a system that it works for each individual cancer, but also kind of works cohesively as a whole. So you're not redesigning everything for every cancer, which is so that's been a bit a challenge as well.
Sure. So moving on about training and learning new things. So how do you onboard people on something like OpenSpecimen? Do you have specific training modules created, or do you do it one-on-one? And in general, how do you keep updated on the latest developments in biobanking?
So at the moment, we're building a series of for guides to help get people up to speed and what to do. We're still in the process of adding new specimens. We had our first organoids come in a few days ago. So we're trying to work through the process ourselves of what does this process look like, and then we can try and build the documentation to try and to capture that process and make it easier in the future.
But generally, it's been we have a very small group of people that can access it and use it at the moment, and the goal really, I think, is to just get that group of people really as competent in using it, and then we will sit down and write some very detailed guidelines about how to use it, and then hopefully we can do some of the putting some of the samples into OpenSpecimen at the different sites. Currently, we're not really confident to do that yet. We want to make sure that it's all running smoothly and working properly for us here. When we can go to Ashlin whenever we need to and say, oh, what do you do with this? Or this isn't working, because it's just it just takes a little bit of understanding, because we're trying to do quite a complex task. There's a lot involved, and so we've decided just to keep it with a small group to start with, and then we'll broaden it with very specific SOPs as we go along.
Yeah, so that brings to the next question about certifications, regulatory compliance, and things like that. So do you have any goals for say ISO or whatever is the equivalent things, or anything else similar?
Yeah, look, we have looked into this before, and not in relation to the biobank, but in reality, trying to certify a research lab is very, very hard, and I really think it's beyond what we want to do. I don't necessarily think there's a need for us to do it. However, having said that, the digital environment that we're using that belongs to that Iridio have installed for us and that we're sort of partnering with them to customize for our needs does have ISO certification among many other certifications. So they have really worked hard on generating those around their platform so that it can safely handle clinical. It can actually, at Great Ormond Street Hospital, it is handling identified information within the clinic. So they have built a lot of security around different parts of their platform, and it's all certified. Yeah, so we're happy with that, and that's really where all of any sensitive data might be. So we're very pleased that they've done that, and we'll just leave our lab as a research lab, I think, where we don't have that type of certification.
Right. So coming to the last question, what's next? So is there any project in the pipeline that's coming up in general? What would be next big thing that?
Yeah, so we have a couple of other cancers that we're focused on in Perth at Perkins. One of the other areas that we're very focused on is women's cancers, and we've been talking now for a few months with some of the clinicians that are involved with hard to treat breast cancers, so triple negative breast cancer, as well as ovarian cancers. So I think, and we have some funding around those cancers as well to start working and collecting those sorts of samples. So hopefully in the next couple of months we'll really start focusing on those. But really, with our opportunity with the liver cancer is really about building a precision medicine database. So we're really keen to continue focusing on just collecting as much clinical information as we can, and really building an evidence-based medicine database that the clinicians can use in the clinic, that will have a really, an integration of all of the research data with all of the clinical data across a patient's entire cancer journey, so that they can then access this information to help them treat other patients when they come in. So that's really one of our main areas, one of our biggest focuses at the moment. And do you want to talk? And Ashlin's going to talk about the Federated Cancer Database.
Yeah, so we're one of our partners is Mindaru, who are one of the projects is the Federated Data Cancer Network. So the goal eventually for us is to put our data into a common data model. So we're looking at the OMOP data model at the moment, and then we can, that makes it easy to federate and share with other people, so that opens up other people to more easily access our data, but also we can access their data as well and query the data, and while still protecting and owning our own data.
And this really has really big implications on the forms in OpenSpecimen.
Right.
So when Ashlin is setting up, it has these all of these conditions and vocabularies that he has to be mindful of. And actually, one of the biggest issues that we're finding is that the clinicians call things, every clinician will call something different, the same thing different. And then there's this federated vocabulary that is totally different, and they don't know what it is. So all of that is actually posing quite a big challenge for Ashlin, but he's managing it really well.
So we've got the clinicians are asking for one set of data, and then we need to try and configure it so the nurses can actually actually enter the data as well, so it makes sense to them. And then this data needs to make sense to the researchers, and then in a couple of years from now, the data also needs to make sense in the in an OMOP format, so it makes sense to everyone. So trying to keep everyone happy has been a bit of a challenge, and we won't know if we got it right until we get to the end.
That's something that's, I think, an universal problem that everyone keeps struggling with in terms of standardization, harmonization, and all kinds of stuff. Okay, so that's pretty much the end of the what I wanted to cover. Is there anything else that you think that we missed that you wanted to bring up?
No, I think that's pretty much covered it. Thank you. It's been really nice talking to you. We're very pleased that we have put OpenSpecimen into our biobank. It's really, it's not an easy task. Opening up an Excel spreadsheet and putting it in that is very easy, but I can absolutely already see so many benefits of actually using a platform like OpenSpecimen. It's helped us immensely, actually.
Yeah, when people ask me who are your competitors in the market, I always say it's Excel.
Yeah. and it still is. It absolutely still is.
Yeah, probably it has like 95% of the market share.
Yeah. But I tell you what, this is just so much better. You just feel more confident with your data that you haven't sort of got a line out or a column missing or something. It's just yeah. So it's a really amazing product for us. We really enjoy using it.
Yeah, using Excel before we moved to OpenSpecimen, and yeah, I pick OpenSpecimen any day.
Yeah, no, it's fantastic.
That's good to know, and that's a good note to end this this podcast and interview. Thanks so much, Louise, Ashlin. It was a real pleasure getting to know, and for me the most important thing is I personally through these 4 sessions I've learned a lot more than probably the last 10 years. So just getting to know, just getting to ask these questions has been very educative.
Thank you. Really appreciate it.
Thanks.